Hi guys, welcome to Fortune YouTube channel. Please like, share, subscribe to our YouTube channel. You can share to your colleagues, to your friends, to any medical students that you know. We're talking about histology today, and note that the slide codes are not peculiar to each slide. They'll be covered during your exam. So first question here is name the cytoplasmic organelle in the organ, and that's mitochondria. How do you identify mitochondria? Is oval shaped, it is small or rod shaped, and then it also have double membrane. And when you look at it, it has tiny granules inside, tiny eosinophilic granules, those blue dots that you're seeing inside of it. That makes it to show that it is what mitochondria, and is abundant in muscle cells like skeletal, cardiac muscle cell, liver cells, and also in renal tubules. Here you have an Golgi apparatus. That's the an organelle that is being stained in this organism. Golgi apparatus. One of the ways you identify Golgi apparatus is that it has this negative staining. There is a part of it that appears unstained. So it has a pure negative staining zone near the nucleus. So if you look at it, you see a part of it that is unstained, and you find it mostly in secretory cells like goblet cells, like plasma cells. Here you have the fat cells. So when you look at the fat cells, you see there's like a large empty looking cell. Like it looks large and it's looking empty also. It is large and empty looking. So when you look at it, you are going to see that there's like a rim of cytoplasm, because it's the bulk of fat cells is made up of fats, is made up of lipid, and then the nucleus is also pushed to the periphery, and that's what results in in what we call signet ring. And you can find it mostly in subcutaneous tissue around organs and also bone marrow. Here you have glycogen granules. You find them mostly in liver cells. Those are hepatocytes, and also in skeletal cells. And one of the ways to identify them is that they look foamy. They have foamy cytoplasm. If you look at it, it looks foamy. And then they also have fine cytoplasmic granule. They have fine cytoplasmic granule. Here you have melanin. So those are the brown granules. So that's one of the ways that you identify melanin. It appears like a brown granules. It's like dark brown or even black sometimes, and you find them mostly in the basal layer of the epithelium. You can find them majorly in the skin, that's the epidermis of the skin, hair follicles, and also retina. Here you have the image of fat cell. Remember, I told you about how it's the way fat cells appear. That it has like a thin rim of cytoplasm, and the nucleus is pushed to the periphery, and looks like a large empty looking cells. So it looks large, but it also looks empty. So if you look at it, you can see that empty space. That those black dots, those are the nucleus. And here you have the simple squamous epithelium. The way you identify simple squamous epithelium is that it has a flattened nucleus, and it's a single layered flat cell. You talk about squamous cell is flat, is wider than it is tall. That's what you say about squamous epithelium, that it is wider than it is tall. So they are made up of flat cell, a single layer flat cell. That's why it's simple squamous epithelium. And then you can find them in Bowman's capsule, in the alveoli, in blood vessels, the endothelium of blood vessels. Here is another image of simple squamous epithelium. So remember, flattened nucleus, they have a single layer of flat cells. Here's another image of simple squamous epithelium. And here you have simple cuboidal epithelium. So simple cuboidal epithelium, when you're talking about cuboidal, is as wide as it is tall. So it looks like a cube. So it's cube shaped, and the nucleus is centrally located. The nucleus is centrally located in simple cuboidal epithelium. Where can you find simple cuboidal epithelium? You can find them in kidney tubules, in thyroid follicles, and the next here is also simple cuboidal epithelium. Though the image is not that large, but that's an image of simple cuboidal epithelium. And here you have simple non-ciliated columnar epithelium. They are taller than they are wide. That's one thing you see about columnar epithelium. They are taller than they are wide. So their nuclei is found at the base, and then it may have goblets, like among them, you are going to find goblet cells. Mostly, you find them in stomach, in intestine, where there is secretion of mucus. So they also have no cilia. That's a simple non-ciliated columnar epithelium. And here also you have the another image of simple non-ciliated columnar epithelium. So you can see the image, it is taller than it is wide, and the nuclei is at the base. Those very thick red spots, those are the nuclei, is found at the base, and they are non-ciliated, no cilia. So here is now simple ciliated columnar epithelium. How do you identify them? They are tall cells and they have cilia on their apical surface. They have cilia on their apical surface. So you can find them in uterine tubes, in small bronchi. That's where you can find simple ciliated columnar epithelium. This is non-keratinized stratified squamous epithelium. So how do you identify it? They have many layers. That's what stratified means. And you remember squamous, they are flat, and then their surface cells are nucleated. When you look at the surface cells, you are going to see nucleus, and then they have a moist surface. If you can feel it, that's moist surface. So you can find them in the esophagus, in vagina, in oral cavity. That's where you can find non-keratinized stratified squamous epithelium. This is also non-keratinized stratified squamous epithelium. And here you have keratinized stratified squamous epithelium. Unlike non-keratinized stratified squamous epithelium, keratinized stratified squamous epithelium, the cells on the surface layer, they are dead and they do have nuclei, and they have thick keratin layer. So you can find keratinized stratified squamous epithelium in the epidermis of the skin. That's where you can find it, in the epidermis of the skin. Here you also have another image of keratinized stratified squamous epithelium. So this is now transitional epithelium. They have multiple layers. It's not really clear here, but that's one thing about them. You see multiple layers, and the cells are dome shaped. The surface cells, they are dome shaped, and they have like an umbrella. So you can also call them umbrella cells. So the shape changes when they stretch. You can find them in urinary bladder, in the ureter. They will change shape from cuboidal, from columnar to cuboidal. So they change shape. So that change in shape, that's why they are called transitional epithelium. And here you have a pseudostratified ciliated columnar epithelium. So they appear to be multi-layered, but all the cells touch the basement membrane. Like the way they look is as if they are different layers, but the difference is that some cells are taller than the other. That's why it looks stratified. So it's a fake stratification. And then you also find cilia, they are ciliated. You find cilia on their on apical surface, and then the nuclei is at different level. This is one of the reason why it's also like a fake stratification, because the nucleus is at different level. So because the nucleus is at different level, it look like it is stratified, whereas they are not stratified. It's just one layer of cells ordering. Here you have elastic fibers and fibroblasts. One of the ways you identify elastic fibers is that they are thin, they are branched, and they are also dark. They are thin, they are branched, and they are dark. So all those dark spots that you see, those are elastic fibers. While for fibroblasts, they have spindle-shaped nuclei. Spindle-shaped nuclei. That's how you identify fibroblast cells. So you can find them in elastic ligaments, in large arteries like the aorta. So you can find elastic fibers there and fibroblasts. So this is collagen and elastic fibers. So collagen is actually thick, unlike elastic fibers. Collagen is thick and it is wavy and pink in color. It's thick, wavy, and pink in color. Why for elastic fibers, those are thin. As I said initially, they are thin, they are branched, and they are dark. So you can find them in the dermis, in connective tissues. Moving to muscle cells now. The first that you have here, okay, let's talk about reticular fiber. So reticular fiber is actually a fine branch, like it has fine branching network, and the best way to see it is when you stain it with silver, so it has like this mesh-like appearance, and that's what you can see, a mesh-like appearance. So this is collagen fiber. This is now muscles. So the way you identify striated muscle is that there is cross striation, visible cross striations on them. If you look at this image, you can see the cross striation on them. So light and dark bands, and you can find examples of striated muscles are skeletal and cardiac muscles. Skeletal and cardiac muscle. And here you have skeletal muscles. How do you identify skeletal muscle? You see that they are long cylindrical fibers. They have long cylindrical fibers, and they also have multiple nuclei on their periphery. They have multiple nuclei on their periphery, and they have strong striation on them. Next is the cardiac muscle, and the cardiac muscle, they are branched. When you see cardiac muscle, they are branched and they have single nucleus and uninucleated, and they have intercalated these dark lines that appears, and then they also have striation. Remember, there are two striated muscles, cardiac muscle and skeletal muscles, and you can find them in the myocardium of the heart. Thank you for listening. Please like, comment, share, subscribe, and thank you.